Hello, welcome to Biology, the scientific study of life, hearing and equilibrium in mammals, as in most other terrestrial vertebrates. The sensory organs for hearing and equilibrium are closely associated in the ear. This figure explores the structure of these organs in a human ear. Hearing. How does the ear convert the energy of pressure waves traveling through air into nerve impulse that the brain perceives as sound? Vibrating objects such as a blocked guitar string or the vocal cords of a speaking person create percussion waves in the surrounding air. These waves cause the tympanic membrane to vibrate with the same frequency as the sound. The three bones of the middle ear transmit the vibrations to the oval window. The membrane on the cochlea's surface, when the one of these bones that steps vibrates against the oval window, it creates pressure waves in the fluid of the cochlea. The waves travel through the vestibular canal, pass around the apex tip of the cochlea, and continue through the tympanic canal, dissipating as they strike the round window. Pressure waves in the vestibular canal push down on the cochlea duct and basilar membrane. In response, the basilar membrane vibrates up and down, and its hair cells alternately brush against and are withdrawn from the tectorial membrane. That causes the hairs to bend first in one direction and then the other with each vibration, as shown in this figure. Notice that. Increasing bending on one direction depolarizes the hair cells, increasing neurotransmitter release and the frequency of action potentials in the sensory neurons with which they synapse. These neurons carry sensations to the brain through the auditory nerve. Bending of the hairs in the other direction hyperpolarizes the hair cells, reducing neurotransmitter release and the frequency of sensations in the auditory nerve. If sound is represented by changes in the frequency of sensations in the auditory nerve, how is the quality of sound conveyed? Two important sound variables are volume and pitch. Volume, loudness, is determined by the amplitude or height of the sound wave. A large amplitude sound wave causes a more vigorous vibration of the basilar membrane, a greater bending of the hairs on hair cells, and more action potentials in the sensory neurons. Pitch is a function of a sound wave's frequency or number of vibrations per second, expressed in hertz, Hz. High frequency waves produce high pitched sounds, whereas the low frequency waves produce low pitched sounds. Healthy young women or humans can hear in the range of 20 to 20,000 hertz. Dogs can hear sounds as high as 40,000 hertz, and bats can emit and hear clicking sounds at frequencies above 100,000 hertz, using this ability to locate objects. The cochlea and distinguish pitch because the basilar membrane is not uniform along its length. It is relatively narrow and stiff at the base of the cochlea near the oval window, and wider and more flexible at the apex. Each region of the basilar membrane is most affected by a particular vibration frequency. The sensory neurons associated with the region vibration must vigorously at any instant send the highest frequency of action potentials along the auditory nerve, but the actual perception of pitch occurs in the brain. Axons in the auditory nerve project into specific auditory areas of the cerebral cortex according to the region of the basilar membrane in which the signal originated. When a particular site in our cortex is stimulated, we perceive the sound of a particular pitch. Equilibrium. Several of the organs in the inner ear of humans and most other mammals detect body position and balance. Behind the oval window is a vestibule that continues to chambers called a utricle and saccule. The utricle opens into the three semicircular canals that complete the apparatus for equilibrium, as shown in this figure. Sensations related to body position are generated much like the sensation of sound in humans and most other mammals. Hair cells in the utricle and the saccule respond to the changes in head position with respect to gravity and movement in one direction. The hair cells are arranged in clusters, and all of the hair project into a gelatinous material containing many small calcium carbonate particles called Atalav's ear stones, which means because this material is heavier than the endolymph. Within the utricle and saccule, gravity is always pulling downward on the hairs of the receptor cells, sending a constant series of action potentials along the sensory neurons of the vestibular branch of the auditory nerve. This mechanism is similar to how statocysts function in invertebrates, and indeed the utricle and saccule are considered to be specialized types of statocysts. Different body angles cause different hair cells and their sensory neurons to be stimulated. When the position of the head changes with respect to the gravity, as when the head bends forward, the force on a hair cell changes and the cell increases or decreases its release of neurotransmitter. The brain interprets the resulting changes in impulse production by the sensory neurons as a change in the position of the head. By a similar mechanism, the semicircular canals arranged in the similar mechanism in three spatial planes detect changes in the rate of rotation or angular movements of the head. Angular movements of the head. Notice this figure. Notice that. When you spin around, the equilibrium in the semicircular canals is disrupted, which is why you become dizzy. Hearing and equilibrium in other vertebrates. Like other vertebrates, fishes and aquatic amphibians also have inner ears located near the brain. There is no cochlea, but there are a saccule, utricle, and semicircular canal structures homologous to the equilibrium sensors of human ears. Within these chambers in the inner ear of a fish, the movement of annelids stimulates sensory hairs. Unlike the mammalian hearing apparatus, the ear of a fish has no eardrum and does not open to the outside of the body. As shown here in this figure, vibrations of the water caused by sound waves are conducted through the skeleton of the head to the inner ears, setting the attalids in motion and stimulating the hair cells. The fish's air-filled swim bladder also vibrates in response to sound and may contribute to the transfer of sound to the inner ear. Some fishes, including catfishes and minnows, have a series of bones called the Webrian apparatus, which conducts the vibrations from the swim bladder to the inner ear. Most fishes and aquatic amphibians also have a lateral line system along both sides of their body. Notice also the system contains mechanoreceptors that detect low frequency waves by a mechanism similar to the function of the inner ear. Water from the animal's surrounding enters the lateral line system through numerous pores and follows along a tube past the mechanoreceptors. The receptor units, called neuromasts, resemble the ambulae in our semicircular canals. Each neuromast has a cluster of hair cells whose hairs are embedded in a gelatinous cap. The copula water movement bends the copula, depolarizing the hair cells and leading to action potentials that are transmitted along the axons of sensory neurons to the brain. This information helps the fish perceive its movement through water or the direction and velocity of water currents flowing over its body. The lateral line system also detects water movements or vibrations that are generated by other moving objects, including prey and predators. In terrestrial vertebrates, in inner ear has evolved as the main organ of hearing and equilibrium. Some amphibians have a lateral line system as tadpole, but not as adult living on land. In the ear of a terrestrial frog or toad, sound vibrations in the air are conducted to the inner ear by a tympanic membrane on the body surface and a single middle ear bone. There also is evidence that the lungs of a frog vibrate in a response to sound and transmit their vibrations to the eardrum via the auditory tube. A small side pocket of the saccule functions as the main hearing organ of the frog, and it is this outgrowth of the saccule that gave rise to the more elaborate cochlea. During the evolution of mammals, birds also have a cochlea. However, as an amphibians, sound is conducted from the tympanic membrane to the inner ear by a single upon the steps. The senses of taste and smell are closely related in most animals. Many animals use their chemical senses to find mates, as when male silk moths respond to pheromones emitted by females to recognize territory that has been marked by some chemical substance, as when dogs and cats sniff boundaries that have been staked out by their spraying neighbors, and to help navigate during migration, as when salmon use the unique scent of their streams of origin to return for breeding. Chemical conservation is especially important for animals such as ants and bees that live in large social groups. In all animals, chemical senses are important in feeding behavior. For example, a hydra begins to make ingestive movements when it detects the compound glutathiones, which is released from pre-captured by the hydra's tentacles. The perceptions of gustation, taste, and olfaction, smell, are both dependent on chemoreceptors that detect specific chemical in the environment. In the case of terrestrial antimales or terrestrial animals, taste is the detection of chemicals that are present in a solution, and smell is the detection of chemicals that are carried through the air. There is no distinction between taste and smell in aquatic animals. The test receptors of insects are located within sensory hairs called sensilla, which are located on the feet and in mouth parts. These animals use their sense of taste to select food. A testing hair contains several chemoreceptors, each especially responsive to a particular class of chemical stimuli such as sugar or salt. Insects are also capable of smelling airborne chemical using olfactory hairs usually located on the antennae, like this figure. Notice that. Okay, notice that. Closer look. Okay, thanks for listening.